Hello there, I'm Peter Zeidman. Thank you very much for joining me. This talk is about dynamic causal modeling for fMRI, and it's all about brain connectivity and how to estimate it using fMRI. I'll start off by talking about different kinds of connectivity, brain connectivity, that you'll read about in the literature. Before introducing the example that I'm going to use throughout this lecture, which is investigating how somebody with a stroke to their language areas of the brain was still able to speak. And I'm going to show you an example using this framework called dynamic causal modeling, DCM. I'll talk about the framework generally, and this is relevant whether you're doing fMRI or MEG or EEG or different modalities. And then I'll come on to the specific DCM models that we use for fMRI. I'll then come back to that stroke example again and just show you what the results look like and what we can learn with this kind of methodology. And finally, we'll talk about group analysis and how you go from the single subject to the group level using something called parametric empirical Bayes, or PEB, which is a kind of random effects modeling. So let's start off with thinking about the system that we're trying to understand. So we've got some time series that we measure with our MRI scanner. But it's often the case that we don't just work with those time series directly. We might do some processing to transform them into other things. So for example, I could average the time series over trials if I was doing an EEG or MEG experiment, and that gives me what's called the ERP, the event-related potential. Or I could look at it in the frequency domain and plot a spectrogram of that called induced responses. Over in the fMRI world, what you'll often find is that people might take time series from different parts of the brain and correlate the time series to generate what's called functional connectivity, correlation matrix, for which bits of the head are correlated with each other. And if they do the same thing in the frequency domain, it's called cross-spectral densities. It's exactly the same thing. What I want to try and get across today is that whilst all of these data features are really important, they are the data that we work with, they're really important, but they are not interesting. What we're interested in as neuroscientists is what's going on in the brain that caused these signals. For example, I might be interested in a particular kind of synapse, and I might have some patients who have a particular synaptopathy, and therefore they have disordered communication between different kinds of neurons. And I might have a hypothesis that a drug improves the functioning of these connections. So how could I possibly go from all of these data features that I measure to asking interesting questions and testing interesting hypotheses about the underlying neural connectivity? Turns out the only way to do that is to have models. Models that say, if the brain were organized in this fashion, what data would I expect to measure? And then, once I've got that data and I've got the model, I can run the model in reverse. I can start with data and ask what was the underlying connection strength, for example, in a particular patient. That's called model inversion, going from consequences to causes. Dynamic causal modeling, DCM, provides the toolkit for doing this kind of modeling, the forward problem and then the inverse problem. And it's important to say, and we'll see this again in a moment, the vocabulary that people use for this, if they're talking about the correlations or other statistical dependencies between fMRI time series, they call that functional connectivity. The really interesting thing I'd suggest is the connections at the neural level. That's called effective connectivity, because it's the effects that neurons have on each other. And so the point of this lecture is to get to the point where we can infer the underlying neural connectivity that caused our data. So let's just recap these different kinds of connectivity. And what you'll find in the literature are three main categories. The first is structural connectivity, and that's the physical architecture of the brain. What's physically connected to what? And the most typical approach you'll find to measuring that is what's called diffusion weighted imaging. And it can produce very beautiful plots like this. But just because two brain regions share an anatomical connection doesn't necessarily mean they're talking to each other. To look at that, you'd have to look at functional connectivity. And as I've already said, functional connectivity is just the correlations or other statistical dependencies among bits of the brain. And because correlations or covariances, that kind of thing, are not directional, there's no arrows on these connections. So it's just correlations, essentially. And if we're doing fMRI, it's the correlations between the bold observations. Effective connectivity are the causal relationships, the effects that brain regions have on each other, and necessarily that you will find arrows on those connections because these connections are directed. The dynamic causal modeling is a framework for inferring the causes of neuroimaging data. You've already learned a lot about the SPM pipeline, so let's see where it fits in. You start off by collecting some functional MRI data, doing the image reconstruction. Then you do your pre-processing, collate all the data together. Then you do your SPM analysis. The SPM analysis shows you these nice colorful blobs for where in the brain were their experimental effects. Having asked where the connections, or where the effects happened, the next question is, why did they happen? So what was the underlying neuronal architecture that caused those blobs? So to find that out, we extract time series from all the brain regions we are interested in, and then we use DCM as the very final step to investigate or to ask what connections could explain that fMRI data. So let me now give you an example from stroke research that I did many years ago. This was my very first piece of academic work when I joined neuroimaging as a master's student with Kathy Price here in London. And this is actually still a piece of work that I'm really proud of today. I think it's quite a nice paper. It's a paper all about something called pure alexia. That's a condition where a patient, usually due to a stroke, loses the ability to read, but they can still write. This study was investigating one patient who had the kind of brain damage you would associate with pure alexia. So she had damage to this part of the brain called ventral occipitotemporal cortex, VOT, highlighted here in green, which the textbooks were saying is an essential part of the language network, the reading network. The thing is, patient AH could still read words so long as they were really short, really common words like cat or dog. And the question was, how is patient AH still able to read despite that lesion? And what you're seeing here in red are the parts of the brain that were more activated in controls. And you're seeing that there may be, there's quite a big language network, but quite a large area of that part where the lesion occurred was lost by this patient in their stroke. So how was she still able to read? So Mohammed Segia, Kathy Price, and others collected data. They had a kind of 2x3 factorial design where they manipulated two things. One was this kind of stimulus displayed to the volunteer lying in the scanner. The stimuli were either words or pictures. And the second condition was whether the stimuli involved naming images, naming pictures of a cat or a dog, or reading the equivalent words. And there were also control conditions. So here's an example of what a trial of the experiment looked like. They would see a cue to tell them they had to prepare to do some reading, and then they would see the words, and they just had to read the words in the reading condition, or if there were pictures, they had to name what was in the pictures. DCM is a hypothesis-driven approach. It's not exploratory. So you have to start off by having quite a clear experimental question. So the experimental question here is that there is some other pathway that can explain how patient AH could read. So we have visual input coming into primary visual cortex. Activity, it was hypothesized, flows along these connections through the motor cortex, planum temporale, ventral occipital temporal cortex. So this is where the patient had a lesion down here. And that the activity would flow to these anterior regions representing the lips and the larynx in motor and premotor cortex. So if the patient had a lesion down here, the hypothesis was perhaps there was another pathway, maybe a dorsal pathway, that could explain how she could still read. And that's what was being tested here. So let's see how you would test that hypothesis. To explain how DCM works, let's start by thinking about the thing we want to model. So we have some stimuli. The stimuli could be words, or as shown here, some little dots on the screen that we are showing to the volunteer who's lying in the scanner on a computer screen. And we know the timing when we displayed those stimuli, on, off, on, off. That causes some change in neural networks in the brain, the effective connectivity, and we can't measure that directly. But what we do measure are the observations, the bold responses. And we've got that measurement, and I'll call that vector y. So let's think now we've defined what's called the generative process. Let's now think about the generative model that's going to correspond to this biological process, biophysical process. So the model is going to start with the experimental stimulus U and end at the observations Y. And it's going to explain what the transformation is in between. And it's going to do that in two parts. The first part is the neural model. The neural model says, how does brain activity Z change over time? So in this simple example, there's just 4 brain regions. And let's say we're going to encode the level of neural activity in each of the 4 regions by a single number. So we've just got 4 numbers. We'll stack those 4 numbers on top of each other and call that vector Z. That's the state of the brain at a given time. The neural model says, what's the rate of change of Z? So this little dot means rate of change or time derivative. So how does Z change over time? Well, it's going to be explained by a model, which I've called here F, which takes in 3 things: the current state of the brain, the timing of the experimental inputs, and this quantity here, which are the parameters, the neural parameters. And these parameters are the really interesting values. These, for example, encode the strength of these connections shown in yellow, or the sensitivity of particular brain regions to the input coming in. That's the stuff that we want to estimate from the data, and that's called parameters. So that's half the model, but we're missing half. Because remember, what we measure with bold fMRI is not neural activity. What we measure with bold fMRI is hemodynamics. So we need a second part of the model. Second part that says, what would we see in the scanner given the neural model? So it translates between neural activity and predicted observations. So our observed data Y, we're saying, is equal to a number given, or set of numbers given by a model, which I'll call G. And it's as a function of 2 things: the current level of neural activity and some parameters that encode hemodynamics. So for example, the rate of blood flow through the vessels in the brain. That would be an example of a hemodynamic parameter, which is also going to get estimated from the data. So that's the complete model then, from start to finish. Why is that useful? Well, it's useful if we can use it to ask questions. To ask questions, we first need to perform what's called model inversion, also called model estimation. In SPM, that uses a particular algorithm called variation order plus, which I'm not going to go into today. But I am going to tell you what its outputs are, because these are the things that could be useful to you as an experimenter. The first thing is it's going to give a probability distribution over the parameters. This reads the probability over the parameters given the data and the model. So remember from if you've already done the Bayes lecture in the course just before this, you will know that the posterior probability is what we believe about something, in this case the parameters, after seeing the data. And this illustration shows what the posteriors look like in DCM. It's what's called a multivariate Gaussian distribution, or a multivariate normal distribution. And if you imagine that these two axes here are two parameters, two connection strengths, for example. So this is the strength of one connection in the brain, and this is the strength of another connection. What DCM gives you is a distribution, or a mountain, over those two connections. Practically, that means what you've got is both the position of this mountain, so what it thinks is the expected value or the estimated value of each connection, but you've also got the width of the mountain in each direction, and the width tells you how confident or how precise that estimate is, and that's really useful for doing statistics. The other thing you get as an output is an approximation of something called the model evidence. In fact, this should say the log of the model evidence, which is the probability that I would have seen my data if the model were correct. Now, this quantity, the model evidence, also called the marginal likelihood, is at the core of all parametric statistics, classical, frequency, or Bayesian. Marginal likelihood is always the quantity that tells us how good a model is. How likely, let's say that the model gave a prediction that looked exactly like my data, then this is going to be a high value. The probability of my data given the model would be high. If my model predicted something that looked nothing like my data, then this would be much smaller. So it's a statistic that tells you how good your model is. Turns out you cannot estimate that precisely, but what you can do is you can approximate it, and that approximation is called the free energy in statistical physics. It's called the ELBO, evidence lower bound, in machine learning. The one thing I want you to take away from that is, well, firstly, that it's just a number. So you can compare different models, and you can ask whether one model is better than another simply by comparing the free energies, by asking which F value is the most positive. More positive is better in DCM. The other thing I want you to take away is that the evidence, as well as the free energy, can be decomposed into two terms: accuracy minus complexity. So a model with a high free energy is either really accurate, or it has a very low model complexity, or some mixture of the two. Complexity here, just to be clear, means how complicated the model is, and practically that means the effective number of parameters in the model. It's a little bit more complicated than that, but that's a simplification. So what we really want when we're modeling is to find the model that offers this best trade-off between being really accurate and not being very complicated. In other words, being really simple. And that trade-off is described by the free energy. And that's why we use it in DCM. How would you use it? Well, let's say I've got two hypotheses. One hypothesis is that my data were generated according to the top diagram here, where there's connections in the dorsal and ventral regions of this brain network. My second hypothesis is that there is direct connectivity between the dorsal and ventral streams. How could I evaluate which of these is the best explanation? Well, I would fit each model to the data. I'd perform model inversion or estimation, get the free energy for each one, and ask which one had the higher free energy. That's called model comparison, or Bayesian model comparison, asking which model best explains the data, and that is the whole point of DCM. It's doing these model comparisons. So, to summarize this section, the DCM framework starts with embodying each hypothesis in a model, and you'll hear the word generative model. Generative model just means that the model generates or simulates the data that you'd expect to see if the model was correct. And importantly, that model separates neural activity from hemodynamics from noise. We then perform a model estimation or inversion that gives us those two useful outputs: the parameters of the model, actually the probability distribution over the parameters. So we could say there's like a 90% confidence that this connection is excitatory rather than inhibitory, for example. And it also gives us the free energy, which is a score for the quality of the model. We then inspect the estimated parameters and/or we compare models to see which model best explains the data. And that's conceptually DCM in a nutshell. Okay, now let's talk about the specific models for fMRI, because everything I've said so far has been quite generic. Let's now get into specifics about fMRI. So here's what we're going to arrive at. This is the neuronal model that's used in DCM for fMRI. Remember, the neural model asks the question, how does brain activity Z change over time? That may look a little complicated, but we're going to go through it and it's all going to make intuitive sense. You can either derive this in terms of mathematics. So for anyone watching this mathematically minded, this is what's called a second order Taylor series approximation of any nonlinear neural dynamics. We're not going to motivate it that way. We're going to motivate it more kind of intuitively and build it up in terms of neurobiology. So let's do it that way, but I'll give some references at the end that you can read to look at the more mathematical derivation. So remember our kind of language network. Let's reduce it down to just two brain regions. Ventral occipital cortex, where every time there's a trial, every time there's three words displayed on the scanner screen, you get a drive coming into occipital cortex. And in this model that we're going to specify, it drives ventral occipital temporal cortex, which can also then send a feedback connection. There is a modulation of this connection, so it can get stronger or weaker, depending on whether the volunteer is reading words or alternatively naming pictures. And the key thing we want to get at is an estimate of these connection strengths, so we can then compare them between the patient and the 20 controls. So let's simplify it down even more to just a single brain region. Let's just take ventral occipital cortex, one brain region. Let's say that we're going to use one number to summarize the level of neural activity in that one region. I'll call that number Z1. We're going to have driving input coming in every time there's a stimulus, and there's also an inhibitory self-connection, which essentially makes sure that this brain region doesn't become epileptic. So it ensures that the activity in that neural region, in that neural population, will decay back down to rest. What would that look like? Well, U1 is the driving input. So this is the input coming in whenever there's a stimulus. So we know the times at which we stimulated the brain. So we can make this vector that encodes when those stimuli occurred, when the trials occurred. Zeros everywhere here, values of 1 every time there's a stimulus. What would we expect the modeled neural activity to look like in an occipital cortex? On average, across all the neurons, you can think of this like average postsynaptic firing rate. Well, we'd expect there to be an increase in neural activity followed by a decay, an increase and then a decay. That may look familiar to anyone who's done any modeling before. This is simply an exponential decay. And we can model that using a very simple equation. So the solution to that exponential decay has this form. It says that the rate of change of Z1, this variable that contains the level of neural activity, is equal to some parameter called A multiplied by the current value of Z at the current time, plus some parameter called C multiplied by the value of U1 at the current time. 0. And this will be a 0 or a 1, depending on whether there's stimulation. So we don't know values A and C, but we can estimate them from the data. Value A controls the rate of decay. And it's important that A is always negative in sign, because that means that this is going to come back down to baseline. C weights the importance of U1 in driving the network. So we call this the driving input parameter. If it was 0, it would mean the network is totally insensitive to the stimuli. And if this was very big, it would say the stimuli have a huge effect. So this C is controlling the height of each of these responses. So the DCM framework, if we just specified this model, could estimate A and C for us in this very simple dynamical system. But of course, it's not very interesting because there's only one brain region, and we're interested in connectivity. So now let's add the second brain region and extend that model. So we've got visual cortex and we've got ventral occipitotemporal cortex now. And let's say we've got a connection, just for this example, in one direction. I'm now going to quantify the level of neural activity in this new brain region as Z2. We've still got Z1, which was the activity in the other brain region. And every time there's a trial, activity is going to flow along this connection to VOT. So we've got a few more parameters now, and I've slightly changed the numbering. A21 is the connection strength going from region 1 to region 2. So you read the numbers back to front, from region 1 to region 2. These other values, these other parameters in purple here, are the self connections. So how much self-inhibition is there in each brain region? So how would we adapt the model we just saw to capture this relationship? Well, the formula for occipital cortex is exactly the same as you saw before. That's what we've already introduced. The self-decay multiplied by the current level of activity plus the driving input. We now introduce a second equation to represent VOT. So that the rate of change in VOT, that's Z2, is equal to a self-decay term plus the level of activity in primary visual cortex multiplied by the connection strength from from region 1 to region 2. So the relevance, if you like, of occipital cortex is now weighted by the strength of this connection. And that's quite a simple equation. What would that look like in terms of predictions, predicted data, or predicted activity? Well, Z1 would look exactly the same as it did before. Z2 might look like a slightly smooth version of it, but exactly how it looks depends on the setting of these parameters. So it gets really clunky if you've got lots of brain regions having to write out all those separate equations. So what you're seeing here is a much more compact version of writing the same thing. What we're saying here is that the rate of change in each of the brain regions, Z, is equal to the connectivity matrix A multiplied by the current level of activity Z plus the driving input. You need to know about this because you need to know how to read this matrix here, in particular the connectivity matrix or the adjacency matrix. To read this, the columns are the outgoing connections and the rows are the incoming connections. So to give an example, A21 is the connection from region 1, so it appears in the first column. So it's the first column is the outgoing connections from region 1. And it targets region 2, the second region in our model. So it appears in the second row. So this is the A21, the connection from region 1 to region 2. You might think about, why is this one here 0? What would 0 correspond to in this diagram? Well, it's the connection from the second region to the first region. And it says, so second to first. So there's no connection in this just little example here, although that would be quite unrealistic. But that's saying there's no connection from region 2 to region 1. So this is a much more compact way of writing it. And we can write that in a simpler form, just collapsing it down, that the rate of change in each brain region is equal to matrix A times the current activity plus the C matrix, that's this, the sensitivity of each brain region to its inputs, multiplied by the input that's coming in. So that's almost the entire model. There's just one more thing we need. It's possible that we want to capture that this connection, or any of the other connections, might get stronger or weaker during our experiment. For example, if you're in the naming condition versus the reading condition, something like that. For that, we just need to slightly append the model. So let's say we've got the input here coming in for reading. Coming into this connection, we say it's modulating this connection. In other words, making it stronger or weaker. That's what we hypothesize. That reading, for example, might strengthen this connection. For that, we need a new parameter, which we're going to call B. B is how much the connection changes due to reading. B from region 1 to region 2. And we're going to have to tell the model when reading occurred during the experiment. So U1 is what we had before. That's every time there's a trial, every time there's a stimulus, there's a little spike that comes into the visual cortex. And what we're now saying is that there was a block during the experiment when people were told to read, and all the other times they were not doing reading. What would that look like in terms of the predicted neural activity Z? Well, Z1 for primary visual cortex will look exactly the same as before. It rises and it decays, it rises and it decays. What about region VOT? Well, because of that boosting effect of reading, if that B parameter was positive, it would mean that that connection is getting stronger during reading and then coming back down again. So these are really powerful connections or parameters because they encode the change in connectivity due to each experimental condition. That's ultimately the most interesting thing for us, usually, in a cognitive neuroscience experiment. How do we model that mathematically? Well, it's almost the same formulas as we had before, but just with a slight addition. The formula for occipital cortex is exactly the same as we had before. The rate of change is equal to the self-connection multiplied by the current level of activity plus the driving input. VOT now, the formula is the same as before, but now it has this extra term. We're going to have this extra modulatory input that says that the input coming in from visual cortex is now going to be modulated up or down, multiplied up or down by the modulation here. And that's the model. If we put all this together in a shorter form, if we compile it more succinctly, then this is what you have. You have that you have the rate of change, as we've seen, in each neural population is equal to 3 sets of parameters in the model, A, B, and C. A is the structure of the network, in other words, which connections are connected to each other once. B is the change in those connections due to each experimental task, and C is the driving input. And in the formula, there's a little summation symbol here because we're summing over the M experimental conditions. If there were 5 experimental conditions, we'd have 5 B matrices. And ultimately, that's it for the neural model. We went through that in a lot of detail. We're not going to go through the observation model in the same level of detail. Remember, the observation model translates from the predicted neural activity to the predicted observations. And the reason we're not going to go through it in so much detail is because it's not something you practically have to change or alter when you're doing your analysis. So I'll just tell you very briefly what this part of the model does. And here you can see it's written on marble to emphasize that it's really important. But it pretty much just works out the box. So how it works is that you have your neuronal input that causes a change in vasodilation. So it dilates the blood vessels, which induces a blood flow. And that flow causes 2 things. It causes the volume to increase. So the actual amount of blood in the vessel increases. And there's also a concurrent change in the deoxyhemoglobin ratio. And together those 2 things pass through a nonlinear function that gives you the predicted bold response. All you need to know is that if you put into this model the predicted neural activity, you get out the predicted bold response. And that forms the second part of the DCM model. So that was a lot of theory. Thank you for staying with me. What I now want to do is show you how that theory translates into results. So I'm going to go back to the example I used at the beginning of a stroke study. And if you remember, we left off where we introduced the patient who has a lesion to this part of ventral occipitotemporal cortex. She could still read very short words. And the question was, how is she still being able to do it despite that lesion? So the pipeline we followed is that we specified DCMs, these models, for every subject, both for one patient and each control. We fitted the models to the subject's data. That fitting is called model estimation or model inversion to give two outputs, a posterior probability distribution for each parameter. In other words, we get a normal distribution or a bell curve over the parameters. And and parameters, for example, would be the connection strengths, A, B, and C. And we also got an estimate of the model evidence. That's this quantity that tells you how good the model is. And because you can't calculate that exactly, we approximate it using something called the free energy. We then chose the models that had the highest free energy or the highest log evidence. And then we looked at those best models and we inspected their parameters. I'm going to skip the model comparison and just show you the parameters. And it's, I think, nicely summed up in this figure. So what being shown here is the brain network we looked at, visual cortex, planum temporale and superior temporal sulcus, motor and premotor cortex, and ventral occipitotemporal cortex, this part where there is a lesion in the patient. Remember I said this is probabilistic. So what you get for each connection is a probability distribution. The black ones here are the connections that were stronger in the patient compared to the controls, and the grey ones are the ones that were stronger in the controls relative to the patient. So we can do a little sanity check. Why would we imagine, if we look at VOC to VOT, why would we imagine that the distribution here for the patient was strongly overlapping with 0? Well, the answer is the patient had a lesion there. And so this fits with what we expected. There isn't evidence to say that they were using that connection. It was much stronger and more positive in the controls. But what we do get are other connections that were stronger in the patient. So VOC to STS is shown here, and you can see much, much stronger in the patient. I should say the axes here are connection strength versus probability density. So much stronger in the patient than in the controls, suggesting that the patient was able to read because she had stronger flow of activation along this dorsal pathway, making up for her ventral lesion. I think more as well about why the width of these distributions is as they are. So why does the distribution for the patient, is it quite wide? And why is the one for the controls quite narrow? Something to think about there. So that's an example of how we could get real insight into the circuitry and the networks of the brain using DCM, comparing a patient to some controls. Since that study was published, a framework was introduced to DCM for doing group analysis. And just in the last 5 minutes, I'm just going to very briefly introduce that. You'll see this mentioned in a lot of papers. It's called parametric empirical Bayes, or PEB. PEB is just a fancy way of saying hierarchical linear regression. What happens is that the connectivity parameters are taken to the group level. So from all our subjects, we collate all of their estimated connection strengths, and they're modeled using a Bayesian general linear model. And Bayesian here means that we're taking into account not just the estimated connection strengths, but also the confidence or the precision over those connection strengths. So what the software will do is it will take this long list or vector of all the subject's connection strengths and specify a general linear model. Your decision as the experimenter is what to put into this matrix X, the design matrix. So very similar to how we were doing an fMRI analysis, a GLM SPM analysis, where you have regressors encoding all your experimental effects. Now you have a design matrix encoding everything that you think might cause people in your population to differ from each other, your sample of the population, rather. So this is just an example. Let's say I've got my just a single connection from 6 different subjects. I'll explain it in terms of these 3 regressors. The first, in fact, always in the PEB software, the first regressor is always a column of ones. That so just white here means 1, black means 0. So a column of ones encoding what is the group average connection strength. So the parameter that corresponds to the first column will be the estimate of the group average connection strength. Here, for example, I might have a group membership, patient or control. So for the second column, I've got -1s and 1s, encoding patient or control, or you could have 0s and 1s. Doesn't really matter. And the third regressor here is some continuous regressor, maybe the effect of age. So with what this will do, what the DCM framework or the PEB framework will return to you are two things. It's going to give you one free energy for the entire group level model. So remember, free energy is a single number, a statistic, that says how good the model is. You'll now get one free energy for your entire population of subjects, taking into account both the individual DCMs and this second level GLM. That's really useful because it means you could try different GLMs, different regression models with different mixtures of covariates and see which gives you the highest free energy. The other thing it gives you are the group level parameters. So these theta2 parameters here, which are encoding the effect of each covariate on each connection. If you have 10 connections per subject, you'll get 10 times 3 parameters here, for example. And that's, I haven't got time to go into more detail now, but there are tools in the SPM software that allows you to do this kind of analysis. So to summarize, DCM is a framework which enables us to make inferences about the effective connectivity of brain regions, which we can't directly observe. And I explained what effective connectivity is. It's directed connectivity at the neural level. We create one or more generative models. Remember, a generative model is a model that can generate simulated data, and then which you can then compare to the real data. We create one or more generative models, each expressing a hypothesis, and we invert those models, or we estimate them using Bayesian inference, which gives us both an estimate of the coupling parameters, the connectivity parameters, and the log model evidence, the free energy. And finally, we compare different models using the free energy. That process is called Bayesian model comparison. And with that, I'm going to finish up. And if you'd like to read more, my colleagues and I wrote quite a detailed two-part tutorial paper with sample data. The first one covers the within subject analysis, and the second covers between subject analysis. So with that, thank you very much.